So we're going to be discussing the thyroid synthesis and utilization pathway and how it's going to affect our metabolism as well as our regulating our stress levels. And so for the first thing that's going to happen is that we're going to have TSH sent from our hypothalamus going down the cellular level. It's going to allow for the transport of sodium and iodide to come into the cell, do this NIS transporter. Now, we have this happening, we're going to be able to bring iodide directly into our colloid through this transporter called pendrin. We're also going to have tyrosine allowing our thyroid globulin to be created in the follicular cell and be brought over into our colloid. Now, something really cool is going to happen here is called iodinization, where we're going to be able to utilize this TPO enzyme and combine it with iodide and create iodine. So iodide is turned MIT and iodine is turned DIT. And so we're also going to have TPO combined with thyroid globulin to allow MIT and DIT to bind right here to our thyroid globulin. And then something that's also really cool is going to happen is called coupling. We're going to be able to combine MIT and DIT to create pre-T3, and we're going to be able to combine DIT and DIT to create pre-T4. Now, keep in mind it's called pre-T3 and T4, so this is going to be brought over from our colloid and exocytosed into our follicular cell through an endosome. Now, this endosome is going to combine with a lysosome and create an endolysosome, where this is actually going to break down the backbone, the thyroid globulin backbone, and create just T3 and T4. This is going to exocytose into our blood, and we're going to have TPG protein, bring it to the bring it to our cell, where we can convert the rest of our T4 into T3. T3 can go directly into the nucleus of our cell to change transcription. Now, what it's going to do specifically is that it's going to increase our oxygen utilization by increasing our number of mitochondria. This means that it's actually going to decrease our stress reactivity. But something that's scary that's going to happen is if we have our HPA axis in effect, so we're utilizing cortisol, we're actually going to be creating RT3 instead of T3. Now, RT3 is going to take up those spots of our T3 receptors on our cells and is going to block it. And that means that we are not going to be able to increase utilization, we're not going to be able to breathe better, and it's going to very severely damage our metabolism.